Welcome to the third PowerPoint of Unit 8, Cross the Line. We have a quote from Neil deGrasse Tyson. Part of what it is to be scientifically literate, it's not simply, do you know what DNA is or what the Big Bang is? That's an aspect of science literacy. The biggest part of it is, do you know how to think about information that's presented in front of you? Our learning goals to understand the nature of diversity in genetics, explain chromosomal mutations, and define different types of dominance. We already know that different alleles with different dominance, that DNA can mutate, and that things really aren't simple. What we saw in the lab is that we did that virtual lab, there's a wide variety of chromosome patterns in people. Things aren't always as expected. We can have different numbers of chromosomes, different problems with chromosomes. Things keep happening. The patterns that we've described so far are very effective at making good predictions, and that's the hallmark of good science. Good science can predict things, and those things happen. The more reliable and the more precise and the more specific you can be in predictions, the better science you're doing. That's what science is. But sometimes there's details. So, for example, we've talked about dominant and recessive. Sometimes there are situations where it's not that clean. You can have things like incomplete dominance. Like in flowers, the dominant R might be red and the recessive R might be white. But if you're heterozygous, it doesn't necessarily mean that the dominant R will completely dominate the white, but you'll get a blending pink. So in incomplete dominance, you end up with your heterozygous, your phenotype, being somewhere in between, mixed together, a blend of your homozygous dominant and your homozygous recessive. We also have something called codominance. In codominance, we see both traits being exhibited. For example, in this flower, we can see that it has some parts red and some parts white. They didn't blend and become pink. They were just at different parts. Sometimes you got red and sometimes you got white. They're both being expressed. We see the same thing with some blood cells problems. Both traits being expressed. Now, a lot of traits we have are polygenic. Poly meaning many, gene meaning the genes we're talking about, meaning that there's actually multiple different kinds of alleles that code for the traits. For example, eye color. Eye color is encoded by two different genes, represented here by A and B. Two different genes, all those patterns come out to 5 different kinds of colors. So for polygenic, it's the combination of multiple genes that determines what the total phenotype is. Unfortunately, nothing's ever quite as easy as we want it to be. Some traits can be described by tons of genes. It depends. Here's a developing question. In this picture, do you think this is an analogy talking about incomplete dominance or codominance? The picture here is trying to get us to talk about incomplete dominance. We can see it blending together. Wonder how long it took to get those dogs sitting together. Though the fact that there's a lot of different shades might suggest there might be some kind of polygenic, but clearly they're making us think about codominance. Okay, what about this example? Talking about blood types, whether you're blood type A, blood type B, blood type AB, or blood type O. This is an example of codominance, because you look at the shapes, the A is represented with triangle shapes on the cells, B is represented by circle shapes in the cells, AB has both, not a blend, both. Or in the case of recessive, neither. Okay, so from the previous slide, let's do a Punnett square for a cross between AB and O parents. Interestingly, in this case, we have 50% of kids with A and 50% of kids with B. We actually have neither of our offspring, neither chance. We don't have any chance of any offspring looking like the mother or looking like the father. It's interesting. If you had to make a prediction, what kind of dominance do you anticipate happens in skin color? Well, it's probably incomplete because we do know that we see some blending. We don't have patches, that would be codominant. So we think it's incomplete, but it's also probably polygenic because we see so many different kinds as well. Now, the genotype isn't always the only predictor of what the phenotype will look like. The phenotype might also depend on the environment. Some animals express different genes based on temperature. There's any number of things. The proof of this is you can take two identical twins, they don't necessarily have all the same phenotypes. For example, one could be taller than the other. Maybe one had a different diet than the other. We also see many traits linked together. People that commonly have trait A also commonly have trait B. That's because that the locus where they're found on the chromosome is near each other. So if you get that chromosome, you're getting both trait A and B as it is. It's interesting whenever it gets to sex linked, though. What's interesting is, it's particularly interesting for males. Sex linked means that the locus for that gene is on the X chromosome. Well, for males, we only get one X. So since males only get one X chromosome, if you get a recessive X chromosome, your phenotype is the recessive. So, for example, for color blindness, that's why it's so much more common for males to have color blindness than for females. Because for a female to be colorblind, they'd have to have two X recessive alleles, but a male only has to have one because males don't get a second X. There's not a second chance to get the dominant. So males are much more likely to exhibit recessive traits in sex-linked examples. Now, males only have one X chromosome. We get by just fine. Females have two, but actually each cell only uses one at a time. The other one gets inactivated. And it's actually random per cell. It happens pretty early on after the zygote phase, and that one stays inactivated for every cell division thereafter. And it's random whether the X they got from the mother or the X they got from the father. That's actually the example of why calico cats are always female. All calico cats are female because what's happening is that the fur color is sex-linked. So the cells that got one color on the X are the orange patches for this adorable kitten, and the ones that are black are the other X. So you get one or the other. In males, you won't see that because they only have one X chromosome, so they all have the same X. We've already talked about how we can get small changes happening at the sequence level, at the nucleotide level. We talked about that in units 5 and 6, but we also can get them happening at the chromosomal level. For example, we get something called crossover, that the two homologous chromosomes, it happens during prophase 1. The chromosomes can switch traits. They're so close to each other, they overlap, and they end up disconnecting, reconnecting. And if you look at the anaphase example, they've swapped with each other. You also get insertion and translocation. The top, we're seeing insertion. So you can see that there's a part of what was on the blue chromosome that's now in the green chromosome. So you're getting information moving around. Translocation. Translocation is kind of what we saw with crossover, where two chromosomes trade little bits with each other, except in crossover there were homologous chromosomes. In translocation there are non-homologous chromosomes. But you can even get within a chromosome. You can just delete a part. You can duplicate a part. So in the first one we're seeing deletion. Just part of it's gone. In number 2, there's duplication. We get that same part in there twice. And then in 3, we get inversion, flipped upside down. You can also have a problem with the number of chromosomes. Sometimes you have too many, monosomy. Sometimes you have, too few is monosomy. Too many is trisomy. You can have extra chromosomes or too few. All of these can cause dramatic problems. Okay, one mastery question. Why is it important to a species that we do have some level of mutation happening? And it definitely doesn't work like X-Men. Because you need some mutations, so you have some random variants, that you have changes and new traits showing up. That way when new environmental situations happen, part of the population can have a trait that'll adapt to it. It's part of the evolution.